Okay, so we'll just wait for 1 2 minutes and then we'll start the lecture. Okay. Okay, a very good morning to everyone present here. So this is the last class, week 13, for this course, that is Introduction to Developmental Biology. So up till week 12, we have completed the entire syllabus, right from week 1 up till week 12, where we have covered starting from the basics of developmental biology to the basic cell biology or cell signaling, which is required in developmental biology, to covering the basic genetics, and then coming to Drosophila development, switching to plant developmental biology, and lastly, also we also studied about, just a minute. Yeah. And lastly, we also studied about the mammalian development. So these were the topics that we have covered in this particular course, and this is just a revision class where I will be where we will be solving questions that we have already solved. Okay, I'm not taking any new questions now because I think now your exams are also coming. Just a minute. Yeah, good morning, Ajay. So now, because your exams are also going to come, I think in just few days. So, uh, in spite of taking some new topic and some new questions, I think it would be better if we start with the already existing. I think it would be better if we start with the already existing questions that we have so that we can revise it. Okay. So I'll be going. Yes, Ajay, you can ask your question or you can just type it in the message box, whatever query you have. Ajay has raised a question. So whatever is your query, you can we get solution of quizzes posted? So quizzes as in the quiz that were given to you by sir for this particular course. So no, I cannot give you the solution of those quizzes because I'm not allowed to do so. Those are the questions that you only have to solve. So that's why we have a doubt clearing sessions, because so that questions which are a little bit similar to the ones that are given to you are, we can solve those questions. So the questions, no madam, I think no madam, after the quiz is over. Okay, so I'll talk about this with the NPTEL team because even I am not clear about it, that we are allowed to do so or not. So I don't know because I haven't been told about this, that we can give the solution of the quizzes which have been posted. So I'm not sure about it, Ajay. Sorry. But I think you can contact the course instructor. Um, if the course instructor can give you, then it would be better, or you can directly message to the NPTEL team. I think NPTEL team would be in a better position to answer than me. Okay, so you can message the NPTEL team. Yeah, so that's what I'm saying, that you can message the NPTEL team and they can probably answer you in a better way. Okay, you can just mail them. They are very responsive. They will surely reply to your mail. Okay, so now we'll starting the questions. So these are the questions that we have already solved. So I hope that you remember the answers and the solutions as well. So we are just going to revise what we have actually learned in this 4 months of duration, whatever topics that we have learned. So the first question is, a researcher wants to study the development of melanocytes. Which of the following germ layers would yield the cells of interest? Okay. A. Ectoderm. B. Mesenchyme. C. Mesoderm. D. Endomesoderm. And E is endoderm. So what should be the answer? I'll give you 1 minute. Today I won't give you 2 minutes because these are already solved questions. So after 1 minute, I'll switch the screen. You can type your answer in the message box. Okay. Okay. Now I'm going to switch the screen. Okay. Oh, myself, ectoderm. So Madiha has answered ectoderm and Ajay has answered ectoderm. So what you're saying, Ajay, myself and many requested same many times. Okay, so that means you have already. Okay, I can also pass the message from my end to the NPTEL team, but rest it is, you can understand it's all in their hands. But I think if NPTEL team is not responding, although they respond, they are very good in responding to the messages. But even if they're not responding, I think you can just try to contact the course instructor. Yeah, yeah, definitely, Ajay. Don't you worry. I'll pass on your message to the concerned people. Okay. So, okay. So the answer that Ajay has answered is ectoderm. So what was the question? A researcher wants to study development of melanocytes. So yes, the correct answer is ectoderm. Okay, because melanocytes are nothing but the melanin, which is a pigment, melanin producing cells. So which are present in the skin. Okay, which is responsible for the coloration of the skin. And people who do not have proper amount of melanocytes, they are also more prone to skin cancers. So melanocytes, in a way, it while providing a pigment to a skin, it is actually protecting the skin layer also. So yes, the correct answer is ectoderm. Okay. Now moving on to the next question. If you have to create a fate map only by direct observation of the developing embryo, which of the following features would you choose? A. Organisms having a small number of cells. B. Organisms having many non pigmented cells. C. Organisms having large sized cells. And D. Organisms having a small number of differentially pigmented cells. Okay. So please note we are talking about fate map by direct observation. Okay. It is written fate map by direct observation. So you have 1 minute to answer the question. Okay, now I'm switching the screen. So Ajay has answered D, that is organism having small number of cells and differentially pigmented cell. And Madhya has also answered the same, that is small number of differentially pigmented cells. Yes. So this is the correct answer. So both have given the correct answer. Okay, so here it is written that you have to create a fate map by direct observations. So before coming to this question, I'll explain this question. Let us quickly revise what is fate mapping. So what is fate mapping? Fate mapping is a technique to trace the cell lineage in developmental biology. Okay, so that means if you want to trace the cell lineage, that from where the cells are coming, what were their ancestors, what was their whole developmental process, for this we use the fate mapping. Now, it helps to understand that how embryonic cells divide, differentiate, and migrate during cell development. That means embryonic cells throughout their developmental process, how have they evolved, how have they divided, how have they differentiated, and ultimately how they went to their required places. This we try to understand with the help of fate mapping. Now, there are different techniques that can be used to study the fate map. First is direct observation. So this question was about direct observation only. So here it was written that if you want to create a fate map by direct observation, then which feature would you choose? Obviously, direct observation means that you are seeing with your naked eyes. So with the naked eyes, if you have non-pigmented cells, pigment means color. So if you have cells which all the cells are of same color, can you differentiate them? No, you cannot differentiate them. So that means you need some kind of coloration where you can differentiate them. Okay, if you have very large number of cells, then also it would be very difficult. That means you need smaller number of cells and which are differentially pigmented. That means the cells are of different colors. Just like in light microscopy we use some dyes, okay, and those dyes gives different colors to the different parts that we want to see, and that is why we can see them through naked eyes. So it has to be differentially colored or differentially pigmented, then only we can see through naked eyes, because it is a direct observation the question is asking. So that is what it is written. So the first technique which is used to study, which can be used to study fate map, is direct observation. Then what is the second one? Vital dye making. Now what are vital dyes? Note, these are the dyes which are given to the living cells. Okay, and these dyes do not harm the cell division or development of the organs. So these dyes are given inside the living cells, but important are that since they are vital dyes, so they do not harm the organs. So this is the another way. First is direct observation. Second is vital dye making. Third is fluorescent labeling. Labeling, that means you can fluorescently label the cells and then you can see them. Then you can use the radio label technique, radio labeling, and lastly genetic mapping. So this is what is fate mapping and what are the different techniques which are used to study a fate map. Okay. Now next question is, which of the following transcription factors can bind to the enhancers even in the heterochromatic? First is strong transcription factor OCT4, B, pioneer transcription factor PAX7, C, potent transcription factor SOX2, and D is pioneer transcription factor PBX. So I'm giving you 1 minute. After 1 minute, I'll switch the screen. Hmm. Okay, now I'm switching the screen. Okay, so Madiha has answered pack 7 and Ajay has answered pioneer transcription factor PBX. So the correct answer are 2. You can see pioneer transcription factor pack 7. This is the correct answer. And pioneer transcription factor PBX. This is the correct answer. Now, important to understand here is that OCT4 can be a strong transcription factor, but to bind to a heterochromatin region, we need a pioneer transcription factor. Why? Because like we have discussed also earlier, that euchromatin is the region which is a transcriptionally active region. Okay, and heterochromatin is a region which is a transcriptionally inactive region. So that means when we say that it is a transcriptionally inactive region, that means that the chromosome is highly condensed in this area. So if the chromosome is highly condensed in the area, then there is no place for the transcription factor to come, sit, bind, and start the transcription process. Since the chromosome is very condensed in this area, that means only those transcription pioneer means first. The first that can come and bind to this particular condensed area so that the condensed area can be little loosened out and ultimately the transcription process can start. Therefore, whenever there is a heterochromatin region, if any transcription factor has to come, it has to be the pioneer. Pioneer means the one which is starting the process. So the pioneer transcription factors, PAC7 and PBX is the correct answer. Okay. Now we'll come to the next question. Which of the following statements is or are true regarding gene and their expression? A is cytoplasmic processing involves the addition of 7-methyl guanine cap at the end of the pre-mRNA transcript. Then cytoplasmic processing involves the addition of a 5-methyl guanine cap at the end of 5' end of a pre-mRNA transcript. The poly A tail is followed by the 3' UTR in the processed transcript. The promoter is always found at the 5' end of the gene and comprises of an multi-rich sequence. Gene silencing involves the methylation of H2A, H3, and H4. The H2A H4 dimer forms a tetramer by binding to the H2B H3 dimer. This tetramer associates with another tetramer to form the core octamer. Then, cytosine methylation is a well conserved mechanism for gene expression. And lastly, transcription factor 2H mediated phosphorylation of tyrosine residues in the carboxyl terminal domain, that is CTD, of the RNA polymerase is required for transcription initiation. So I can understand it's a big question. So I'll give you 3 minutes. Go through all the points, whatever points are given, and then you can solve it. Okay. And I'm speaking a little slowly because I'm in the lab right now, so I cannot be that loud as I used to be in the earlier classes. But I hope I'm audible to you. So, okay, so your time starts now. You have 3 minutes to solve this question, okay? Okay, 2 more minutes left. One more minute left. Okay, now I'm switching the screen. So, most of recent articles suggest that Mayodi and Fox are pioneer, not PBX. Yeah, I think, see, so science always evolves, right? So there are always some conjectures, and then always there are some uh, things that change. So I don't know. I mean, according to whenever the question was framed, maybe then the articles were not, I mean, taken into concern. But if you know that, it's good. That's good for you. Okay. So Ajay has answered except B, all are correct, and Madiha has answered G and E except B, all are correct. Okay. Okay, so no, the correct answer is only one option is correct, and that is G. Only G is correct. Okay. So now I will tell you. The first option is cytoplasmic processing involves the addition of 7-methyl guanine cap at the 5' end of the pre-mRNA transcript. Yes, 7-methyl guanine cap is added at the 5' end of the pre-mRNA transcript. But these are the processing that happens inside the nucleus and not in the cytoplasm. Okay, so this is not a cytoplasmic processing. This is a nuclear processing, and therefore this option becomes wrong. Okay, then the next one is again cytoplasmic processing involves the addition of a 5-methyl guanine cap at the 5' end of the pre-mRNA transcript. Firstly, it is not 5-methyl, it is 7-methyl, and secondly, it is not a cytoplasmic processing, it is a nuclear processing. So this is also wrong. Then the second one is the poly A tail is followed by the 3' UTR in the processed transcript. So yes, the poly A tail is added to the 3' end of the mRNA, but it follows the 3' UTR, not the other way around. Okay. Poly A tail is not followed by 3' UTR, but poly A tail follows the 3' UTR. So this is also incorrect. Then the promoter is always found at the 5' end of the gene and comprises of an 80 rich sequence. So most of the promoters they contain 80 rich sequences. For example, Tata box. Tata box is a promoter and it contains 80 rich sequences. But this is not true for all genes. So we cannot generalize it. So it is a more generalized statement that a promoter is always. No, it should not be always. Okay, it can be mostly but not always. So therefore it becomes incorrect. Now someone wants to join. Yeah. Then the next is gene silencing involves methylation of H2A, H3, and H4. Okay. Now, gene silencing involves methylation. That means here we are talking about what? Here we are talking about histones methylation. Now, histone methylation typically affects H3 and H4, but they do not necessarily affect the H2A histone. Okay, so gene silencing, whenever we talk about, if you know that whenever we talk about any kind of methylation and modifications, we mostly talk about H3 and H4. H2A is not very common, it's not actually involved. Therefore, had it been gene silencing involves H3 and H4, it would be a correct answer. But because H2A is also written over here, therefore this is incorrect. Then the H2A H4 dimer forms a tetramer by binding to the H2B H3 dimer. This tetramer associates with another tetramer to form the core octamer. Okay. Yes, there is a core octamer that is formed. Yes, the two individual tetramers come together to join and form a core octamer. But what are these two individual tetramers? Sorry, yeah, I mean the individual tetramers from the dimers. The dimers are not H2A, H4, and H2B, H3. The dimers are H2A and H2B form a dimer, and H3 and H4 form a dimer. So H3 and H4 form a dimer, and H2A and H2B form a dimer. Not this. Therefore, it is a wrong statement. Then cytosine methylation is a well conserved mechanism for gene repression. Yes, this is true. So we have already discussed this. Again, I'm telling that two kinds of first is methylation, okay, and the second one is acetylation. So acetylation, it promotes transcription, and methylation, it transcription. So, in general, of course, there are certain exceptions, but in general, methylation represses transcription and acetylation promotes transcription. And where methylation occurs mostly, it occurs in the CPG islands. That means at the cytosines. Okay. So the question is cytosine methylation is a well conserved mechanism for gene expression. So this is correct. Then the last one is TF2H mediated phosphorylation of tyrosine residues in the CTD. CTD is carboxyl terminal domain. In the CTD of the RNA polymerase 2 is required for transcription initiation. So is this correct? No. Why? Because TF2H phosphorylates the serine residues in the carboxyl terminal domain, not the tyrosine residues. Although in some cases they do phosphorylate the tyrosine residues also, so it is a little contradictory statement. But according to the answers that have been provided to you, this is wrong. So uh, uh, the most plausible explanation that I can understand from this is that it is phosphorylating the serine residues. Okay, because they are not considering the tyrosine residues. Okay, so this is just you can see this is a cartoon that represents a gene and all the sequences that are present in the gene. This is the data box. This is the promoter region. This is the 3' UTR regions. This is exon, and this is exon number 1. This is exon number 2. And exons are present not in a continuous fashion, but they are interrupted by introns. So these are intron. This is intron number 1. This is intron number 2. So this is just a general cartoon or a pictorial representation of how a gene looks like. Okay, now we'll come to the next question. Hmm. Function of RISC is to find the complementary strand and cleave the RNA, to inactivate the RNAs and protect the RNA from the risk of cleaving, to find the complementary strand and cleave the DNA, and to inactivate the DNAs and protect the DNA from the risk of cleaving. Okay, so these are the four options. You have 1 minute to solve this, and after 1 minute, I'll switch the screen. Okay, you're answering. Okay, 30 seconds more. Okay, so Ajay has answered B. Madiha has answered find the complementary strand and cleave the RNA. Hmm. Okay, good. So the correct answer is to find the complementary strand and cleave the RNA, which is option number A. Now, so Madiha has answered it correctly. So let us try to understand this question by quickly revising the biogenesis of miRNA. Okay, because this will clear all your doubts. So see, the microprocessor complex brosha. So first of all, you have a primary miRNA. The primary miRNA is formed from the miRNA gene. MiRNA, what is the query now? Okay, fine. Let us see this question. So miRNA means primary miRNA. Okay. Pri stands for primary. So first of all, what is happening? So this is your nucleus and this is your cytoplasm. So inside the nucleus, you have your primary miRNA. After transcription, your primary miRNA is formed. Now this primary miRNA, which is miRNA, is acted upon by the DROSHA or DGCR8 complex. This DROSHA, it cleaves the primary miRNA and then it forms pre-miRNA. So from pri, it forms pre. Okay. Then this pre-miRNA comes out of the nucleus. Now, how it will come out of the nucleus? With the help of the exportin complex. So this exportin 5 helps in the pre-miRNA to come out of the nucleus. So now this pre-miRNA comes out of the nucleus. So up till here, whatever was happening, that was the nuclear processing for the miRNA. Okay. And from here, this is cytoplasmic processing. Now, what happens? So, always remember, DROSHA acts during the nuclear processing, and dicer and RISC they are present in the cytoplasmic region. So, always remember this. Now, this pre-miRNA is acted upon by dicer, and then it is cleaved, and it forms two strands. The first one is the passenger strand, and the second one is the mature miRNA strand. So this passenger strand, it is degraded, and mature miRNA functions as a guide. So now this mature miRNA, it goes to the risk complex. I mean, there are two strands. The passenger strand is degraded, and the other strand, uh, after the risk complex, you can see, so degradation happens after the risk complex. Okay, so here you can see there are two strands after the treatment of dicer. There are two strands. First is the passenger strand, and other one is the mature miRNA strand. The other miRNA strand goes to the risk complex, and the passenger strand is degraded. Now this mature miRNA, this is responsible for the uh, uh, repression. The repression can happen in two ways. If this miRNA is perfectly complementary to the mRNA, then it will degrade the mRNA. If this miRNA is not perfectly complementary to the miRNA, it will repress the transcription. So either mRNA degradation can happen, or repression of the translation can happen. It depends upon the base pairing. If the base pairing is perfect and it is perfectly complementary, then it will happen what? mRNA degradation. If the base pairing is imperfect, then the mRNA will not be degraded, but ultimately the transcript translation would be repressed. So this is how your miRNA is synthesized and it performs its work. So first in the nucleus, pri miRNA is converted into pre-mRNA with the miRNA with the help of DROSTA. Then it comes out of the nucleus. Then it is acted upon by dicer. Then after the dicer cleavage, we have two strands. The one is a passenger strand, other is the normal miRNA strand. Passenger strand is degraded. Other miRNA strand, it goes and binds to the mRNA. Whether it is binding perfectly or imperfectly, it will decide whether the mRNA is degraded or only translation is going to repress. Okay, so this is your miRNA biogenesis. So now we know risk. What is the function of risk? To find the complementary strand and cleave the RNA. So now this risk will help in finding the complementary mRNA strand, so that your this miRNA can go and bind to it. What were the other options? To inactivate RNAs and protect the RNA from the risk of cleaving? No, it is not going to protect the RNA. It has to cleave the RNA. So this is completely wrong. To find the complementary strand and cleave the DNA, no, it is not cleaving the DNA, it is cleaving the RNA. To inactivate the DNAs and protect the DNA from risk of cleaving, no, it is not protecting neither DNA nor RNA. It is just cleaving your RNA. Okay. So this is your explanation. Okay. Now, next question is, hmm, if the central VPC is destroyed, what would happen to the development of primary vulval cells? VPCs, vulval precursory cells. They will not be formed. They will transform into hypodermis. They will be formed normally, or they will acquire tertiary fate. Okay, this is your question. And you have just a minute. Someone wants to join. Hmm. This is your question, and you have a minute to solve it. Then I'll switch the screen. Okay. Okay. Now I'm switching the screen. Okay. So Ajay has answered C and Madiha has answered they won't be formed. Okay, let me see. If the central VPC is destroyed, what would happen to the development of primary cells? So the correct answer is C. That is, they will be formed normally. Okay. Now why it should be C? C. The central verbal precursory cells, that is VPC. First of all, we're talking about C elegans. Okay. Then what happens if the central verbal precursory cells are destroyed in C. elegans? The neighboring verbal precursory cells, specifically we're talking about the secondary VPCs, they will undergo a process which is known as cell fate induction. Okay, so the secondary VPC will undergo a process which is known as cell fate induction if the central VPCs are destroyed. This means that one of the adjacent cells can adopt the primary fate. Okay, one of the adjacent cells performs the primary fate due to lateral signaling. So that means if the primary verbal precursory cell is destroyed, the secondary verbal precursory cells which are present adjacent to it, they, due to lateral signaling, they adopt the primary fate. Okay. This adopts the primary fate. This ensures normal functioning of the verbal cells. Okay. So this means they will be formed normally as neighboring VPCs can adopt the primary fate and maintain normal verbal development even if the central VPC is destroyed. Is it fine? So, two things are important. First is cell fate induction, and the second one is lateral signal. Okay, fine. So, I hope this is clear to everyone. Just a minute. We have a lot many questions. Okay, fine. We'll see how much we can cover. Okay. Now the next question is, which of the following chemicals can be used to induce mutation? EMT, EMS, MET, and MES. Okay. So try to answer this question and you can type your answer in the comment box. Okay, so I'm switching the screen now. Have you answered it? It was a simple question. I think you must have answered. Okay, fine. So, um, okay. Madiha has answered EMS. Ajay has answered A, that is EMT. Okay. And Srivat Ravi. I'm sorry, I'm if I'm pronouncing your name incorrectly, but he has answered EMS. Okay. So the correct answer indeed is A, which is EMS. Okay, ethyl methane sulfur. So EMT, is it a chemical? No, EMT is not a chemical. EMT is a phenomena which is epithelial to mesenchyme transition. Okay. So this is EMT. EMS is ethyl methane sulfonate. It is a chemical. EMT is not a chemical. EMT is a phenomena, process, epithelial to mesenchyme transition. So the correct answer is B. That is ethyl methane sulfonate. MED is mesenchyme to epithelial transition. So EMS is the correct answer. Okay. The next question is, in a tissue recombination experiment, which of the following will show lens induction? Okay, so this is the genotype of the optic vesicle. This is genotype of the surface ectoderm. So in accordance to this, try to solve this question. I will give you 2 minutes to solve this question. Okay. 1 minute left. Okay, now I'm switching the screen. Huh. So, okay. So, ma'am, is there a way to access notes after this after class? Yes. So you must have a sheet, okay, where you are given the link of all the recorded lectures of YouTube and also all the PPTs that I have had shared. So you must have that Excel file somewhere that must have been shared to you by NPTEL. So there you can go and search. Or even if you don't find it, I'm writing my name here. Okay. So if you type this in YouTube, you will get my channel name. You will get my channel. So there you can go in developmental biology because there are many other courses also that I have taken previously. So in developmental biology specifically, you can find the recorded lectures. But I am sure that you must have been given Excel, I like some file where you have the links of all the recorded lectures as well as the PPTs. And rest, whatever has been taught by the course instructor, that is already given in the portal. Okay, fine. No problem, Shiva. Now we'll see the question. So, hmm. What people have answered? Shiva has answered B and D, Ajay A, B and D, and Madiha as B. Okay, just remember what. Yeah, I think at the bottom, of course, I think in your portal itself, I think there is some at some place the link should be given. You can go and check it. Fine. So let us try to understand this question. Before, so the correct answer is B and D. That means B and D are the correct ones. So that means pack 6, and wild type, and wild type. Now let us try to understand this question because this question covers an important topic of induction and competence. Okay? See, there are two things that you need to understand. First is inducer and another is responder. Inducer is the tissue that produces the signal. Okay. So there would be a tissue or cell that will produce a signal, and then there would be another tissue that will take this signal and it will respond to this signal. So this tissue which is responding to this signal is known as responder. And this tissue which is producing the signal that will change the behavior of the signal, this tissue is known as inducer. Okay, so there are two things, inducer and responder. Now let us come to this question. In a tissue recombination experiment, which of the following show eye lens induction? So here we are talking about the induction phenomena. That means one has to be the inducer, other has to be the responder. So your optic vesicle is your inducer and your surface ectoderm is the responder. This is your responder and this is your inducer. Fine. Now let us see here. I'll try to, I'll do is that, yeah. Now see here. Eye lens induction will happen if both optic vesicle and surface ectoderm are normal, in their normal form. Then eye lens induction will happen. Okay. When optic vesicle is uh, mutant of PAC6, that means PAC6 is not present in the optic vesicles, but it is present in the surface ectoderm, then also eye lens induction is happening. When optic vesicle is wild type, but surface ectoderm does not have PAC6, it is a PAC6 mutant, then no induction is happening. When both are uh, uh, mutated for PAC6, then no induction is happening. So from which this, what can we get to know? That the requirement of PAC6 at the surface ectoderm is essential for the eye lens induction. See, here also PAC6 was mutated, but in surface ectoderm it was normal. So eye lens induction was happening. But if your optic vesicle is a wild type, but your surface ectoderm does not have PAC6, then eye lens induction will not happen. So this means PAC6 is an essential anterior ectoderm to be able to respond to optic vesicle. That means whatever signals are given by the inducer, which is your optic vesicle, in the responder, which is your surface ectoderm, PAC6 is an essential factor which ultimately responds to the signal which is coming from the optic vesicle. Okay, so from here we get to know that optic vesicle is such an important part over here. So I think this question is clear to you. Now here you can see it is wild type, it is PAC6. So now here the PAC6 is not there. So here lens induction cannot happen. Here it can happen because the, see, you have to look at the ectoderm genotype. If it is wild type, it can happen. Doesn't matter whether this is wild type or mutated. Here you see both are mutated, it cannot happen. Both are wild type, it can happen. So the correct answer would be B and D. Now you see here, if it is written PAC6 and PAC6, two times it is written. What does it mean? It means that it is a homozygous mutation. That means one gene has two alleles and both the alleles are mutated for PAC6. Both the alleles do not have PAC6. Okay, this means not present or knocked out or removed, something like this. And these two individuals that you see, this means in both the alleles, the mutation has happened. Fine. Now, yes, Ajay, you can ask your question. You can just write it in the comment whatever your question is there. Now we come to the next question. If bicoid is added to the middle of the bicoid deficient embryo, which of the following is or are expected expected embryonic pattern? A. Acron, B. Abdomen, H. Head, and D. Thorax, T for Telson. So these are your options. What do you think would be the pattern which is expected if bicoid is added in the middle of the bicoid deficient embryo? Okay. So, Ajay, I have already told you that I cannot discuss these questions. I'm really sorry, but I cannot discuss the questions that have been given to you. Okay, this is the question. I'm giving you 2 minutes. Just think about it and try to solve it. Okay, this is right now it is 11:57. I'll switch the screen at 11:59. Okay, you have 2 minutes to solve this question. 1 minute left. Yes. 30 seconds more. Okay, your time is up. I'm switching the screen now. Hmm. So what you have answered? Let's see. Madam, same question you asked now just about pack 6. Okay, fine, Ajay. Madiha has answered C, Ajay has answered C, and Srivad, Ravi, okay, I'll just call you Ravi, okay? He has also answered C. Okay. Which is Telson, thorax, head, Telson, abdomen, and Telson. Yes. And of course, if this is happening, the embryo is not viable. Okay, so I think you have answered it correctly. So, what happens that in Drosophila development, your bicoid gene is crucial for establishing the anterior posterior axis. That we already know that bicoid gene is responsible for the establishment of anterior posterior axis. Okay, now normally, what happens that in a normal case, your bicoid is concentrated in the anterior part of the embryo, which leads to the formation of anterior structures, that is head and thorax. So, normally what happens? Acron, head, thorax, abdomen, and telson. So, bicoid is present in the anterior region. Now we have an embryo which is deficient of bicoid, and we are adding bicoid in the middle. So what will happen? The structures that are responsible uh, by the bicoid, that is head and thorax, now they will be formed in the center. Okay, so when they will be formed in the center, just a minute. Now that Ajay, whatever they have given, uh, so okay, yeah, okay, I'll take the questions later because it breaks the flow. Just let me explain it first. So now, what happens when bicoid is added in the middle of a bicoid deficient embryo? What it will do? It will induce the formation of anterior structures around the side of bicoid injection. Then, the resulting embryonic pattern, it will reflect the gradient of bicoid in the middle. So always remember, it is actually the gradient of the bicoid that decides what kind of embryonic pattern it will find. Okay, so head will be formed with the highest concentration of bicoid and then thorax a little further out. And then your abdominal and posterior structures are formed where the concentration of bicoid is low or completely absent. So in this way, it would be what? Thorax, head, thorax, abdomen, and here it would be telson. Have I answered it correctly or not? Yeah, of course, there would be telson. So C would be your correct answer. Just a minute. I have something to. And since. Okay. And since this kind of structure has formed, uh, therefore, embryo will not be viable. So, just a minute. Yeah. So, therefore, hmm. So now, why acron is not formed with head in center? Okay. See, acron formation in the interior of the embryo, it depends on two things. It not only depends on the bicoid protein, it also depends on the action of maternal terminal genes. Okay, so maternal terminal genes are the genes which are required for the formation of acron and telson. Okay, and acron, which is present at the terminal head structure, is not present in bicoid mutant because, see, you have to understand it is a bicoid deficient embryo. Now, if it is a bicoid deficient embryo, although we are inducing the bicoid protein later on, but the because it is a bicoid deficient embryo, therefore the maternal terminal gene which works in accordance with the bicoid also gets affected. And because of that, since now the maternal terminal genes cannot function after a particular stage, they convert into the proteins and they were not formed earlier because it was a bicoid deficient embryo. Therefore, acron is unable to form. So, therefore, this embryo is not viable because all the structures are scattered here and there. Okay. Now, the next question is, okay, I'll just increase the size so that you can see. Yes. Identify the A, B, and C in the following figure. So I have taken those questions which cover some of the important topics that we have studied up till now in this particular course. So all the important topics that we have, not all, but at least most of the topics that we have covered in this course. So the questions related to that I have tried to take over here. So this is a question which is related to a signaling pathway. Okay. So you have to identify A, B, C, and D. So what are A, B, C, and D? So your options are: A can be wingless, B can be hedgehog, C can be transcription of armadillo, D can be transcription of engrailed or hedgehog. A is hedgehog, B is wingless, C is transcription of wingless, D is transcription of engrailed and hedgehog. A is hedgehog, B is wingless, C is transcription of engrailed and hedgehog, and D is transcription of wingless. And A is wingless, B is hedgehog, C is transcription of hedgehog, and D is transcription of engrailed. So don't go on the this marking. This is randomly. I think I was doing something. So don't think that this is only the correct answer. Okay. So I'll give you 2 minutes to answer this question. It is 12:07 right now. I'll switch the screen at 12:09. Okay. Try to solve the question. Anyways, we are going to see this pathway also because it is an important pathway. One more minute left. Okay, your time is up. Now I'm switching the screen. Hmm. Okay. So Ajay has answered B. Ravi has answered B. Uh, Madiha has answered B, and Ajay has answered B or C something. I don't know. Either B or C. Okay. Okay. So before answering this question, let us try to understand the pathway. Okay. So you see here in this pathway, you can see that how hedgehog signaling works in coordinates with the wind signaling. Okay, so first we'll start anywhere we'll start. Chalo, we'll start with here. Transcription of the wingless. So wingless gene is transcribed first of all, and when this wingless gene is transcribed, it forms protein which is known as wingless protein. Now, this wingless protein will go and it will bind to the receptor which is present another cell, and it will bind to the receptor which is your frizzled receptor. So this is your wind signaling. Not exactly wind signaling, but yes, this is the whole part of wind signaling. Okay, so in wind signaling what happens? So in wind signaling, if wind comes and bind to the receptor, then beta catenin is not destroyed by the beta catenin destruction complex, and therefore the transcription of the required gene takes place. In very short, I'm telling you what happens in the wind pathway. What happens? There is wind on and wind off. So wind on means wind ligand or wind protein is present and it will bind to its receptor. That is your wind on state. You have a wind on state and you have a wind off state. So wind on state means your wind ligand is present. Wind off state means wind ligand is not present. So this describes the wind on state. What happens? The wind protein comes, it binds to the receptor. And because it binds to the receptor, then what happens? That beta destruction complex, beta destruction complex which actually destroys the beta catenin, that beta destruction complex is deactivated, and therefore the transcription of the respective genes happens. So now, and when the wind is and when wind ligand is not present, this beta catenin is destroyed and therefore no transcription happens. So now we know that this particular pathway requires the wind proteins or wingless proteins. But from where these wingless proteins are coming? So they come because of another pathway which happens to the nearby cells. So from here, the transcription of wingless protein, wind protein happens. So now this will ultimately lead to the transcription of engrailed and hedgehog. So when engrailed and hedgehog are transcribed, just a minute, someone wants to join. Yes, fine. So when engrailed and hedgehog are transcribed here. So this engrailed and hedgehog are transcribed because of this wind pathway. So this hedgehog will come out of this particular cell and it will bind to the another receptor, which is your patched receptor. And because this hedgehog will go and bind to the receptor which is present in the nearby cell, which is your patched receptor, then what will happen? Then, along with this smoothen protein, then it causes the transcription of wingless. So you see, it's a cycle. Wingless is getting transcribed because engrailed and hedgehog has been transcribed, and hedgehog comes, and engrailed and hedgehog are transcribed because wingless is getting bind to the receptor. So it's a cycle. So each and everything is dependent on each other. Okay, I think this pathway should be clear to you. Now let us see this figure. Here you can see. So what is this? This is hedgehog. Okay, this is A is your hedgehog. What is here? What is happening? Here transcription of engrailed and hedgehog is happening. Here, what is this? This is wingless. And what is happening here? Here the transcription of wingless is happening. Okay, so this is your hedgehog. This is your wingless. Here transcription of wingless, and here transcription of engrailed and hedgehog. Okay, this is the pathway. Fine. Now let us come to the next question. Simple question after a detailed question. Which of the following makes it difficult to study mammalian embryonic development? A. Large embryo. B. Fewer zygotes. C. Cleavage is faster than amphibians. Or D. Internal embryonic development. Oh my god, we have questions. Okay. So I'll give you 1 2 minutes to solve this question and then I'll switch the screen. It's 12:16. I'll switch the screen at 12:18. Okay. Fine. Let's try to solve this. We have 2 minutes to solve this question, although it's an easier question. Okay, now I'm switching the screen. Ha. So Ravi has answered D and Madiha has answered B and D. B and D. Okay. So the correct answer is B and D, I guess. Let me see. Yes. So why embryonic development is difficult to study? Because unlike other small organisms in mammalians, zygotes that are formed are less. Fewer zygotes are formed. Then the internal embryonic development, that means whatever the development that happens, the embryo development, it happens inside the body. So it's even difficult to trace it, catch it, and study it, unlike where it happens externally, that means outside. So this is because there are fewer zygotes and because there is internal employment development. That means the development of the embryo, it happens inside the body. Okay. Now, next question is identify the correct statements regarding the ABC model. The genes of the subsequent roles inhibit the genes of the prior roles to form organ boundaries. For a flower to have only petals, loss of function of class C genes is required. Class D gene mutant will be sterile. Loss of function mutation in Sepatella that we'll see will have similar phenotype to leafy mutant, and loss of function mutation in class B genes will feminize the plant. So this is a question which is related to your plant developmental biology and ABC model. So this ABC model is a very important topic, a very important concept. So that's why I've taken this question again for so that we can have a revision of this particular concept. So you have 2 minutes. I'll give you 2 3 minutes to solve this question. Okay, and then we'll switch the screen. 2 minutes more. Now I'm switching the screen. Hmm. How we should know multiple answers are to be given? Zygote is small. You can ask the course instructor Ajay because he is only going to frame the questions and he has only given the answers. I'm sorry, but I'm unable to answer it. You have to see by yourself when you're going through the questions, then you have to see that what can be the answers of the questions. Okay. Okay. Madiha has answered A, B, and E, and Ajay has answered except B. All seems correct. Okay. Fine. So to answer this question, let us first try to understand the ABC model of flower development. Okay. Here you see four. Basically, this ABC model of flower development describes that how the sepals, petals, stamen, or carpel in a flower are arranged. The arrangement of the flower, okay? It describes the arrangement of the flower parts. So there are four roles. First is sepals, second is petals, third is stamen, and the fourth is carp. Okay, so these are the four roles which are present in your flower. And how they are arranged, what is the pattern of their arrangement in normal condition and in mutated condition, all these things are described by the ABC model of flower development. Okay, now you see this is the normal condition. In normal condition, one thing more. These are the two things. This is the first thing. Second thing is that there are three genes A, B, and C, and these three genes control the development of these four different parts. So, how they control? That is what it is being described in the normal condition. Also, A, if A is mutated, then C will come in its place. If C is mutated, then A comes in its place. They are co-dominant in nature. That means if A is mutated, then A replaces C, and if C is mutated, C replaces A. These are the two things that you need to understand. Now let us see. First is sepal. Sepal will form only when A is present. Gene A is present. Petal forms when both A and B genes are present. That means for the formation of sepals, you need only gene A. For the formation of petals, you need A as well as B. For the formation of stamen, you need again B plus C. And for the formation of carpel, you need only C. Okay, so this is how these three genes control the development of the four whorls or the four different parts of a flower. This is your wild type condition. Okay, now we'll see the three conditions one by one where the mutation has happened in gene A, gene B, or gene C. This ABC model is very important. Again, I'm telling you. Okay, now let us see. When mutation happens in A gene, sorry, yeah. So when mutation happens in A gene, then what will happen? If A is mutated, C comes in place of A. So that means now here C will come, here also C will come. So what will happen? Earlier there was only A, now there is C alone. So C alone will form carpel. So here it is carpel. C plus B forms stamen. So it would be carpel, stamen, stamen, and carpel. Okay? Because A takes, when A is mutated, C takes over its place. When C is mutated, A takes over its place. Now, next is when B is mutated. What happens when B is mutated? Then A forms sepal. Here it should be A plus B. Now nothing can come in place of B. So it would be alone A. Here it would be alone C. So sepal, sepal, carpel, carpel. Okay. So here what was happening? Carpel, stamen, stamen, carpel. Why? Because A was mutated, so C came in place of B. Now the last case in which mutation happens in gene C. That means when C is mutated. So what happens? If C is mutated, then A comes in place of C. So here alone A will form sepal. Here A plus B form petals. Here normal condition it should be B plus C, but because C is mutated and A has come in its place, so it is A plus B, which is petal, and then A alone, which is sepal. So what would be the phenotype? Sepal, petal, petal, and sepal. Okay, so this figure essentially describes the ABC model of flower development. I hope it is clear. Now we'll come to the question and we'll try to solve the question. Okay. Yeah. The genes of the subsequent rules inhibits the genes of the prior rules to form the organ boundaries. Yes, it is correct. Why? See, there is regulatory interaction between genes which ensures the proper organ formation because A inhibits C and C inhibits, sorry, uh, yeah, and C inhibits A. So that means that they inhibit each other. There is inhibition that happens, and because of this inhibition only, the proper formation happens. And because A inhibits C, therefore, wherever A is present, C cannot come. And because C inhibits A, therefore wherever C is there, A cannot come. And therefore when A is mutated, quickly C comes and takes over its place. And when C is mutated, quickly A comes and takes over its place. Okay, so yes, this is correct. For a flower to have only petals, loss of function of class C genes is required. So see here, we want only petals. Petals are controlled by A plus B. That means you need to have A and you need to have B both in order to form petals. Here they are saying that loss of function of class C gene is required for a flower to have only petals. So if C is mutated, if there is C is mutated, what will happen? Here C plus B should be there, but A plus B. So here would be petal, petal. But only petal flower is not possible. Why? Because if C gene is mutated, then two sepals will be formed. If A gene is mutated, then I mean, if C gene is mutated, then two petals will be formed. But wherever A alone is present, so sepal will also form. So therefore, only petal flower is little difficult. It is not possible. So this is wrong. Then next option is class D gene mutant will be sterile. Now class D, we haven't studied about class D. So class D genes are involved in ovule development. Okay. Class D genes, they are involved in ovule development. So if class D genes are mutated, then what will happen? The proper development of ovule will not take place. And when the proper development of ovule will not take place, what will happen? The mutant flower will be a sterile one. So this is correct. Then loss of function in septella gene will have a similar phenotype to leafy mutant. This is also correct. So SEP, what is written over here, sorry, SEP stands for Sepaleta gene. Okay. And these genes are required for identity of the floral organs. Okay. This Sepaleta gene is responsible for the identity of the floral organs. And leafy is also crucial for floral meristem identity. So, mutants in both cases result in severe disruption of flower formation, which leads to similar phenotype, that is, flower turning into a leaf-like structure. Okay, so basically, Sepaleta is responsible for maintaining the identity of the floral organisms. So if Sepaleta gene is mutant, then what happens? That the proper formation of flower is interrupted. There is disruption in the proper flower formation. And this leafy gene is also responsible for maintaining the identity of the flower. So in both the mutation cases, the phenotype would be very similar. So this is a correct statement. Then loss of function mutation in class B gene will feminize the plant. Let us see here. Okay. So if B is not there, then it would be sepal. Here also it would be sepal. Here it would be from here you can see, if B gene is not present, that means it is A, and C. That means sepal, sepal, carpel, and carpel. So, is there any formation of stamen that is happening? No, because for the formation of stamen, you need B plus C genes both. But for the formation of carpel, you need only C gene. So if there is a mutation in the B gene, here you can see, here is the mutation in the B genes, then two sepals are formed, two carpels are formed, but there is no formation of stamen, the male part. So what happens? This is correct. That loss of function mutation in B genes, it will indeed feminize the plant. Okay. So this was an important topic to discuss. Okay. Now let us come to the next question. During gastrulation, the cell lose A. N-cadherin, B. E-cadherin, C. P-cadherin, or D. NP-cadherin. You have 2 minutes to solve this question, and then I will switch the screen. Okay? It's 12:36. I will switch at 12:38. After, okay. So now I am switching the screen. So Ajay has answered. Okay. Okay, Ajay is not answered. Okay, Madiha has answered E. Catherine. Okay. And yes, E. Catherine is the correct answer. So basically, what happens is that during gastrulation, there is a process which is known as EMT, which is epithelial to mesenchymal transition. So this EMT process, which is epithelial to mesenchymal transition, it happens during the gastrulation process. So epithelial cells, which are cuboidal in shape, they are changed or transformed into spindle-shaped mesenchymal cells, and therefore there is a kind of redevelopment or the reformation of the whole outer lining takes place. So in this process, E. cadherin is lost because E. cadherin, it plays a crucial role in maintaining the epithelial cells in a tight fashion. Okay, so epithelial cells are tightly bound together because of this particular E. cadherin. So E. cadherin, it helps in tightly binding the epithelial cells. But since the epithelial cells has now to be transformed into mesenchymal cells, that means the connections have to be loosened out because spindle shaped cells which are present, these are little loose cells, they are not so tightly bound cells. Okay, that means E. cadherin has to be lost. So therefore, E. cadherin, which is responsible for the tight bounding of the epithelial cells, is lost during gastrulation. Why? Because epithelial to mesenchymal transition event takes place. That means epithelial cells are converted into spindle shaped mesenchymal cells. Okay, fine. Now we'll come to the fine. Sorry, got DC. Fine, Ajay. Good. Now, next question is, bats retain interdigital webbing by expressing dash in the wing epithelium. A. FGF8, B is BMP, C is Wnt, and D is SHMG. So you have 2 minutes to solve this question. It is 12:40 right now. I'll switch the screen at 12:42. Okay. Okay, I think you have already answered. So now I will we'll see and discuss it. Just give me 1 minute. Okay, let it be. Fine. So Ajay has answered BMP, Madhya has answered BMP, and Ajay has answered FGF8 is also correct. So first of all, let me tell you that both BMP and FGF8 cannot be correct. Okay, either BMP has to be correct or FGF8 has to be correct. We'll see and discuss what can be correct here. So the question is, bats retain interdigital webbing by expressing dash. So the correct answer is FGF8. And why not BMP? Let us discuss it. So FGF8 is your fibroblast growth factor 8. This is your FGF8. Now, FGF8, here it is question says that bats retain interdigital webbing. That means if this is the hand of bats, so this webbing is retained. Okay, this webbing is retained because of FGF8. That is what the answer says. Why? Because FGF8 it inhibits the apoptosis that happens in this area. So FGF8 it inhibits the apoptosis that happens here. So therefore the web pattern is retained. And what is the function of BMP pathway? BMP it promotes apoptosis. So they have contradictory role, totally opposite role. BMP it promotes apoptosis. So that means if BMP is there, then there will be distinct fingers. There would not be a webbed structure because BMP is promoting apoptosis and FGF8 it inhibits apoptosis. So that means FGF8 is inhibiting apoptosis, which clearly indicates that FGF8 is in a way inhibiting the BMP pathway because BMP pathway is responsible for the apoptosis of this region, the cells of this region. Okay, so inhibition of the BMP signaling with the help of factors such as FGF8 allows the retention of the interdigital webbing, which is necessary for the wing formation. Is it clear to everyone that how FGF8 and BMP have a totally opposite, contradictory role? BMP is promoting apoptosis, therefore, it leads to the formation of distinct individual fingers, whereas FGF8 it inhibits the BMP signaling and thereby it inhibits the apoptosis. Therefore, the webbed structure is maintained, which ultimately can lead to the formation of the wings. Okay. Now, next question is given below are some statements regarding mechanisms of evolutionary change. First is sickle cell anemia. It is the last question for the day and for the course also. Sickle cell anemia is caused by heterotypy of beta beta hemoglobin. B. Hemophilia A is a result of heterometry of factor 8. C. Most cancers are caused due to heterometry of P53. And fourth statement is thanatophoric dysplasia is caused by heterochrony of FGFR3. And these are all the options that are given to you, and you have to choose the correct statement. Okay. So I'll give you 2-3 minutes because there are not many options. So just see and analyze all these options and then try to answer it. Okay. It's 12:45. I'll switch at 12:48. So try to solve. So let others also answer, Ajay. I think you can wait for at least 1 or 2 minutes. And whatever query you have, you can just type it in the message box. 2 minutes more. One minute left. Okay, now I'm switching the screen. I think you all have answered it already. Yeah. So, okay, okay. So, whatever question you are writing, Ajay, I'm sure it's not the question that I have mentioned. So, please don't put the questions that are given in your quiz. I cannot repeat it again and again that I am not allowed to discuss those questions. Just try to remember the concept that BMP is responsible for reducing apoptosis in the interdigital skin layer, whereas FGF8 it inhibits the apoptosis process. Okay, so see, you have written BMPs are responsible for interdigital apoptosis. That's what I'm saying. And rest, what they have answered, why they have not given you a correct answer, that I don't know. You can ask the course instructor. I just read. I'm reading what you have written. Hmm. I think you can, whatever queries you have regarding to the questions that are given in your quiz, it's better that you ask the course instructor that why they have not given you marks or whatever it is. Fine. And rest, yes, yes. Sure. If you have a class on 28th, you can definitely compile all your queries, whatever queries you have, and you can ask him because he has only made these quiz questions. He has only made the questions that I am discussing with you, and he's only going to make your question paper. So he would be the main resource person to whom you should contact for the queries. For other problems related to my questions or some concepts that you can ask me, but why the questions are marked incorrectly and why what should be the answer of the questions, that I think yes, the concept should be correct. Exactly, you are completely right. You should definitely ask him whatever your queries have. Okay, now we'll discuss the last question. Okay, so just give me 5 minutes. I have my PI is calling me. Just give me 5 minutes. Hello, Rishabh. Hello, hello. सुनो सुनो हाँ, मुझे तुम मैसेज कर देना और मैं क्लास। ठीक है, ओके ओके। Hmm. Okay. So now, yeah. So you should know first of all what is the meaning of the terms heterotopy, heterochrony, heterotypy, and heterometry. So heterotopy means what? Heterotopy means change in form. Okay. Heterotopy means change in form. Then heterochrony means change in time. Heterotypy, it means change in kind. And heterometry, heterometry means change in amount. Change in amount. Okay. So, yes. So, what have you answered? Let me see. Okay. So, now the correct answer here is first, third, and fourth. First, third, and fourth. These are the correct answers. So, first is sickle cell anemia is caused by heterotypy of beta beta hemoglobin. So, heterotypy means that there is a change in the form. And of course, a change in form takes place. How it takes place? Because the S shaped hemoglobin, which is a flexible, it is changed into rigid sickle shaped uh, cells. So the red blood cells, which were earlier of S shaped, they are changed into rigid sickle shaped cells. So this is a kind of change uh, in the form which is taking place. So therefore, this is correct. Then most cancers are caused due to heterometry of P53. What is the meaning of heterometry? Heterometry means change in the amount. That means the amount of P53 which is formed, that is changed. So yes, because when there is a mutation takes place, so there is a change because there's a mutation takes place in P53. Therefore, obviously it becomes non-functional, and then therefore the amount changes. Then the last is fourth, that is thanatophoric dysplasia is caused by heterochrony of the FGFR3. So heterochrony means change in time. That means the time at which the transcription of FGFR 3 gene takes place, it is changed. And because of this change in the timing of the transcription of this particular gene, this particular disease, which is known as thanatophoric dysplasia, is formed. So you should, for in this question, this is okay, but what is more important here that I wanted to focus on is the meaning of these terms: heterotypy, heterochrony, and heterometry, heterotopy. So these are the terms which are very recurrently used in many of the questions, and many of the concepts, many of the examples are also given in your lectures. Okay, so you have to go through all those examples and you have to see like what is the meaning and in which context they have used those examples. Okay, so this completes the questions for this week and for this course also. So if you have any doubts, you must have you're going to have a online one-to-one discussion with the course instructor. You can ask him. If the doubts are at my level, even I can clear them. You can uh, ask me. You can just type it in the comment section anywhere in any of the YouTube channel, I mean YouTube video. And all the best for your examinations. Hope you do well in your examinations and hope you succeed with good marks. Yes, Ajay, you can ask your question. You can just type it in the message box, whatever question you have. Okay, okay, there were many. Sorry, I couldn't. Okay, please enter just one query. Just tell me is it true or false? No explanation needed. Loss of FGFR results in failure to activate stat1. You can Google it, Ajay. Even I have to also see. So if I tell you something wrong, it won't be correct thing. Okay. So I also would need to see this exactly that it will actually activate stat one or which stat. Okay. So all the best to all of you, and thanks for joining with me in all the lectures. And I wish all the best for examinations. Bye. Hmm.